This paper, titled Long-Read Sequencing of 3,622 Icelanders, provides an insight into the role of structural variants in human diseases and other traits. What is the paper telling us? What does the long-read sequencing give us above and beyond the classic short reads of Illumina sequences?
Yeah, so for the past 10 years or so, people have mostly been focusing on these short reads. And they are able to give a picture of all the smaller variants in the genome, SNPs and indels. And there are parts of the genome where the short reads don't reach that well. Places that are deleted between individuals, places that are repetitive in the genome. And these regions we can now access. So we can access all the structural variants in the genome. And these structural variants are, of course, much larger than the standard SNPs and indel that people commonly look at. And they have a much greater chance of having an impact on the phenotype. So we can see variants that have a very large impact on phenotype and play a truly causative role in the disease.
But still, with the short read sequencing, we could get a lot of structural variants. So what is the quantitative difference between using these long read sequencing technology from Oxford Nanopore versus using the Illumina sequencing?
So we have qualified that there's about 40% of the variants, the structural variants, that simply cannot be found and cannot be genotyped with the short reads. The remaining 60% you can find with the short reads, but often it requires very complicated and elaborate methods, and it can be quite difficult to find these variants. But the 40% you simply cannot find with any algorithms.
In addition to the structural variants, what else does the long-read sequencing give us?
So we haven't discussed this in this paper, but there is the potential for actually getting the smaller SNPs and indels in these repetitive regions of the genome. So there are regions that are multiplied many times, genes that, many important genes that, like the LPA gene, which have a very complicated repeat motif that is multiplied multiple times in the genome. And we can, with this technology, we can get the smaller variants also in those repetitive parts. And the Oxford Nanopore technology also has the possibility of giving us methylation information. So the modifications that are done to the DNA in the cell, so apart from the base pairs themselves.
So in addition to giving you more structural variants, more reliable identification of structural variants, giving you some of these hidden regions of repetitive sequences, it gives you an opportunity to examine covalent modification of the DNA.
Yes.
And you sequenced about 3,600 genomes, but you could nevertheless look at the output from that in a much larger group. How did you do that?
Yeah, so once we have these variant calls in the individuals, the 3,600, we can basically look at the inheritance pattern of those variant calls across the larger set of individuals. And we have over 170,000 Icelanders that are now in our databases. And we can infer how those variants were inherited in that population of 170,000.
So you impute the structural variants into the others, and then you went on, once you had imputed into this 160,000, you started to look at the impact on various human phenotypes, diseases, and other things.
Precisely, yes.
And what came out of that?
We found quite a large set of structural variants that had been correlated with disease already, that had been correlated with SNPs already in a catalog, GWAS catalog of known associations. And now, instead of correlating it with SNPs, we can now correlate this with the structural change in a gene itself. So that means we can directly implicate a particular gene. That was the first finding. A second finding was we found structural variants that are associated with disease. And I think there were a few thousand of those that we found. Some of these were actually deleting or inserted into a particular gene. So we found a variant in that deleted the first 2 exon of a gene called PCSK9, which lowered the LDL cholesterol levels in individuals in the carriers of that deletion. We found another repetitive motif inside a gene called ACAN, and basically the number of copies you have that repeat correlates linearly with the height of the individual.
It's a positive correlation.
Yes.
So the more repeat you have, the taller you are.
Yes.
So is this one time that biology makes sense?
Yes.
So one of the when I look at this paper, when I think about what you have described, it sort of looks like we have still another time, an opportunity to say that we have finally completed the sequencing of the human genome, because you are providing insights into regions that hitherto have not been seen because of the use of sequencing technology that didn't allow us to see them.
Yes. So we're still closing that gap a little bit, and I think made a substantial effort in closing that gap. There are a few parts that are simply so repetitive and so complicated that even this technology doesn't handle them. There are, of course, we have studied this in a large set of individuals, 3,600, which is much larger than anybody has done before. But we'll of course never know the full human genetic variation unless we study even larger sets.
Is it fair to say that what this paper is describing is much more a promise of things to come rather than specific genetic discoveries made and described in this paper, because you have introduced to our population study a new technology that should give us a lot of more correlations between diversity in the sequence and diversity in human phenotypes?
More or not, it is a promise, but we have also made some effort into the correlations. So, but I think yes, definitely there is going to be promise in using this technology better, improving the algorithms, increasing the sample sizes, and better cataloging the sort of genetic variations.
Thank you.